So thank you very much to Labroots for inviting me to share our experience with the clinical development of medicinal signaling cells for liver regenerative medicine. I am Etienne Sokal. I am professor of medicine at UC Louvain University in Belgium, and I am a CEO of a spin-off company called Cellaïon. So the story started quite a while ago. I was, and I am still an hepatologist, a liver doctor, and I am used to do liver transplantation, but liver transplantation is always a bit frustrating because you have to remove the whole liver to replace with a new liver. What we wanted to do is to repair the liver and to restart functioning normally. And this is particularly the case for children with inborn error of metabolism because there is one single missing enzyme and the rest of the liver is perfect. So we started to do what is called hepatocyte transplantation. It means that instead of replacing the full liver, you are just infusing liver cells in the existing organ to bring the missing enzyme activity. And the first one was done in 2001. This was the first hepatocyte transplantation in Europe. And it was in a child with a very, very rare disease, a peroxisomal disorders. But it went well and it was successful. And what we could see in this patient is that there was an improvement of the circulating metabolites. Your liver is really the machinery that is processing everything in your body. This child had a missing enzyme. Because of that, he had abnormal metabolites accumulating and being toxic for his brain, eyes, and ears. And thanks to the hepatocyte infused in his own liver, there was an improvement. And as you see, the colon, there was a first improvement due to the mass of cell between D0 and D6. But after that, there was some kind of relapse due to the disappearance of hepatocytes which were not successfully engrafted. But thereafter, there was a progressive improvement due to engraftment and even proliferation of transplanted hepatocytes. So this was really exciting, and we started, of course, to treat other patients with inborn error of metabolism. These were patients with urea cycle disorder. The urea cycle disorders, you do not process ammonium properly, you do not synthesize urea, and we could see that by infusing hepatocyte, we had an improvement in the urea synthesis. Besides that, we could also demonstrate engraftment in the liver of this patient, and we could see appearance of de novo enzyme activity. We could detect donor cells by FISH technique for the Y chromosome, and we could measure progressive increasing enzyme activity from 0% progressively to 3 to 4%. And this is a huge difference between 0% and 4%. It modifies completely the phenotype of the disease. It brings you to a normal, a near normal phenotype, although you still are at risk of decompensation in severe situation, but no more in a daily life. So that was very successful. The inconvenience is that you need to infuse the cells in the liver of the patient, so you need to access the liver by percutaneous route in the portal vein. This vein is really difficult to access because it is located between the gut and the liver. So that was a bit the most difficult part of the technology. So there were some hurdles, and the first one is that what we infuse is not really pure hepatocyte. It's just a liver cell suspension. You take the liver, you isolate the cells with collagenase, and you infuse a kind of soup of liver cells. The durability is unfortunately not as long as a full liver transplantation. So after 18 months, we started to have a loss of the efficacy of the hepatocyte transplantation. access to the liver tissue is also, of course, limiting the technology. You need more or less to have access to one full liver to treat one recipient. And you do reach few % of enzyme activity. Ideally, you should reach 10 to 20% to have a really full control of the metabolic disease. Access to the portal vein, as I mentioned, is a bit technically difficult. And in addition to that, it is a full GMP process, so you need to have a GMP facility to isolate hepatocyte. So what we are doing, in fact, is now it was in the tissue bank of Clinique Saint-Luc from University of Louvain. We obtained a human liver tissue, and this human liver tissue is perfused with collagenase. You have this soup of cells, and this soup of cells is then used to transplant hepatocyte. But what we have done also as a quality control, we have plated these cells in vitro to obtain culture of hepatocyte. And what we did observe, which was quite interesting and novel, is that the hepatocyte do not persist very long in culture. But what we did observe is the emergence of fibroblastoid cells, and these fibroblastoid cells take over, and it becomes a pure population of mesenchymal stem cells, but derived from the liver, so it's specific mesenchymal stem cells that could be then expanded in vitro, and you have this pure population of cells. And this was an invention because this was not reported and described at that time. So we have characterized these liver stem cells extensively. And our hope at that time was, of course, that these cells could replace the hepatocyte, and that you can expand these cells in large quantities so that with one source of liver tissue, you can treat many, many patients. So these cells are really amazing because these cells are MSCs. They have the same MSC markers than bone marrow or adipocyte-derived MSCs, but they have specific hepatocyte markers like ASMA and albumin. There is no hematological marker and no biliary marker. So they are unique, and they are also unique because they do express simultaneously ASMA and albumin. So this is not a cell that you can find in the body. This is a cell that appears in the process of cultivating the hepatocyte and the progenitor cells. And this was, of course, the originality of the cell. What was also interesting is that the cell, because it's derived from the liver, the cell does not express class 2 HLA. And class 2 HLA is an issue in terms of immunogenicity, and patients can develop antibodies against class 2 antigen, which is, of course, something you want to avoid, especially if the patient will require organ transplantation later. But the benefit of cells coming from the liver is that they do not express this class 2 HLA-DR. So then, of course, we decided to use this cell instead of the hepatocyte to treat patients with inborn error of metabolism. And we did treat first child with this liver-derived stem cells. This was again a child with urea cycle disease, and we could demonstrate 3 to 5% engraftment of these cells in the patient, and there was a stabilization of ammonium concentration and decreased orotic aciduria, which is also an abnormal metabolite in these patients. And the patient did benefit from the procedure with less decompensation episodes. So what was also important is to demonstrate that these cells, when you infuse these cells in the liver, they do persist in the liver. And this was beautifully shown with Indian labeled cells. As you can see on this slide on the right panel, the cell reached the liver after 5 minutes, but they persist up to 5 days in the liver. And this is beautifully shown also in this video. And in this video, you can see the progressive filling of the liver with the cells, like if you would fill a bottle with some liquid, you are just filling the liver of the patient with cells. So that is really amazing, especially also because these cells then do not bypass the liver, they persist in the liver, they do not go out of the liver. And this patient was the first one, but we then started to develop the spin-off company called Cellaïon, and the aim was to treat patients with this disease, with urea cycle disorders. We have treated 24 patients, and we could demonstrate safety of the product. This is always the first thing you have to demonstrate in clinical development, but also that as compared to the observational group, there was an improvement of ammonium level in the blood of this patient. And we had a very sophisticated technique using C13 urea synthesis in this patient. We could demonstrate de novo urea synthesis following infusion of cells in this patient. However, there were some limitations. First, again, the level of improvement was probably not sufficient to compare with full liver transplantation. The second limitation was this intraportal infusion. As I mentioned earlier, it is technically heavy for the patient. You really need interventional radiology to find access to the portal vein. So you can do that once, twice, but you cannot not do that repeatedly. So what we decided to do is to ask a volunteer to receive the cells by peripheral vein infusion. A peripheral vein infusion is really easy to apply. And what we could show, which was really amazing also, is that after infusing the cells in the peripheral vein, the cell will cross the lung, but they will not persist in the lung. This is the normal circulation flow from your right arm to the lungs, and then to the lung, from the lungs to the heart, and from the heart to the liver. And these cells have made the way through the lungs and through the heart, and they did reach the liver. 60%, the green upper line shows that 60% of the cells infused do reach the liver and persist in the liver. The two lines showing the lung very transient entrapment, and then there is disappearance of the cells from the lung. And the in-between lines are quite interesting also. You see the orange one, there is a peak, and this peak corresponds to the right lower panel where you see a hotspot of cells focusing in a joint, because this patient had joint inflammation. So the conclusion is that when you infuse the cells by peripheral vein, they do reach the liver, they persist in the liver, there is some loss of cells in the reticular endothelial system, but there is also a specific homing not only to the liver, but also to inflammatory site. And this is, of course, the next development for this product, because in between, the concept has changed. The cells are not aimed to transdifferentiate into hepatocyte. They are not aimed to replace fully the tissue. But these cells have a very strong and powerful paracrine activity. And this is more and more demonstrated now, including for this specific cell. HepaStem has a strong paracrine component. The cell will produce cytokines, and these cytokines act in three different ways. The first one is a strong anti-inflammatory component, thanks to prostaglandin E2, IDO activity, TGF-beta, and this will fight inflammation in the liver. The second step is to release a hepatocyte growth factor, which will help regenerating the liver tissue, but also sphingosine 1-phosphate, which will block the leakage of the endothelial barrier, which is a component of chronic liver disease. And finally, last but not least, TGF-beta and metalloproteinase, which play an anti-fibrotic role. In addition to that, because we originally treated a genetic disease, we are in an allogeneic setting, which means that with one source of tissue, you can treat many, many patients. And again, it's quite easy to apply now that we can infuse the cell by simple peripheral vein infusion. So what we are doing now is really the cells are cultivated in very large scale in a GMP facility, and they are stored in small vials. These vials are cryopreserved, and then you can ship the cells in these cryo container. You can ship these cells around the world. They are stabilized in this dry shipper for 2 weeks. Then you just thaw the cells, reconstitute with a diluent, and infuse the patient. So we decided then to treat patients with strong and severe inflammation of the liver. And this is unfortunately the case of many patients with chronic liver disease. You can have chronic liver disease even not knowing that, for many reasons. First, if you drink too much alcohol, if you have a fatty liver, if you have autoimmune disease or viral disease. There are many reasons to have a chronic liver disease, but you can feel very well because you do not feel your liver. And then suddenly what happens is that when the disease progresses, suddenly you become jaundice. The patient is yellow. He develops also ascites, which is fluid in the abdomen. And this is typically what is called acute decompensation of the liver. So at that time, it's only the liver. But because your liver is the central organ of the body, because of that, other organ will fail. You will develop what is called multi-organ failure: heart, lung, kidney, brain. And this is what is called acute and chronic liver failure. It is a very severe condition, very life-threatening. If you see on this slide, overall, the mortality at day 90 in yellow is above 50%. And there are different grades of severity, and the mortality is between 40 to 70%. So very severe life-threatening condition, and this is the one we have attempted to treat first. So usually, if you have acute and chronic liver failure, you have jaundice, and the mortality of this patient can even reach in more than 70%, especially if the patient have bilirubin elevated above 5. This is again jaundice. So we started to treat the most severe patient because, of course, this was a new treatment, cell therapy. So we would not develop cell therapy for mild and minor diseases. And the first clinical development is, of course, a phase 2A study. Phase 2A is mainly dedicated to demonstrate the safety of your product. It's always safety first, but also to try to see some preliminary efficacy results, and also to have a dose finding for your product. And what was really interesting in this uncontrolled study, phase 2A, is that we did observe in a number of patients a decrease of jaundice. Decrease of jaundice, but also decrease of the MELD score. The MELD score is the score that gives you priority for transplantation. and a number of patients had improvement of the MELD, improvement of Child-Pugh score, improvement of bilirubin, but also the two last panels show improvement of inflammation, which is the mechanism of action of the cells. CRP was decreasing and leukocyte count was also decreasing. So this was quite encouraging. First, we have shown safety, and we have signs of preliminary efficacy. So we went back to the regulatory authorities with a protocol designed at this time to establish the proof of concept that this HepaStem cell can indeed treat patients with such a severe life-threatening condition. In addition, we also could see that on the long term, there was an improvement in the function of the liver, and this is albumin synthesis. And on the right, that the kidney function was maintained. What is usually the case in these patients is that due to the liver disease, there is progressive kidney impairment, kidney function impairment. And now the designers of the clinical trial is a placebo-controlled study because we want to bring this proof of concept. We treat severe patients, patients with high bilirubin, patients who are persistent acute and chronic liver failure, which means that they must be diagnosed with the disease, but we treat only the ones who do not recover spontaneously. Then they receive 2 infusions of the HepaStem. 2/3 of the patients receive the product, 1/3 receive placebo, and the primary endpoint is at day 90. We want to obtain an improvement of the survival of these patients. So this is an ongoing trial, and we, of course, hope to deliver good results by mid of next year. But it's not the unique possibility because, as I mentioned, the cells and HepaStem is also fighting inflammation. One huge disease around the world is NASH. NASH is non-alcoholic steatohepatitis. It is a complication of NAFLD. NAFLD is non-alcoholic fatty liver disease. At that time, you just have a fatty liver. But then suddenly, and we don't know precisely why, inflammation comes on the top of this fatty liver, and this is steatohepatitis. This is a chronic liver disease, which is becoming now the first cause of liver transplantation in adults. So what we want to do, and what we have just published now, is that HepaStem is able to migrate to inflammatory site, to secrete this anti-inflammatory molecule, and this will help decreasing the inflammation in the liver, and the hope is to revert NAFLD, to revert NASH to NAFLD, to stop the inflammatory component and to avoid the progression of steatohepatitis to cirrhosis. And the animal model was just published these days in Cells, and we are, of course, developing a clinical development program for this indication. So what is nice with cell therapy is that it is a cargo. It's not one unique medication. It's multiple medication contained in a cargo. You deliver the cargo to the tissue, and the different mechanism of action will then act in synergy. And that's why we have a pipeline of different indications. The first one is acute and chronic liver failure. We are in this proof of concept trial. We hope to deliver the demonstration of efficacy by mid of next year. And then we can extend the clinical development to NASH, acute decompensation, alcoholic liver disease also, just to treat any patient who has inflammatory liver disease. And of course, MSC therapy has been in the year 2000. There was a hype for MSC therapies, but then there was some disappointment because at that time the view was to replace tissue. But now there is a huge comeback of this technology because now we know that the mechanism of action is paracrine, anti-inflammatory, regenerative. And this is really why we think that we can now bring the breakthrough that is still expected in MSC therapies. First, because of this strong paracrine component, because we are in an allogenic setting, so the logistic is quite easy. With one source of tissue, you treat many, many patients. Because these HepaStem come from the liver and they return to the liver, and they have low immunogenicity, very simple peripheral vein infusion, no need anymore to do intraportal infusion. In between, we have an outstanding safety profile. We started to treat children with inborn error of metabolism, but now more than 100 patients have been infused with perfect safety profile. We have this preliminary efficacy data, and now we are in this proof of concept study. We had the green line from the Data and Safety Monitoring Board to pursue the study without any change. And of course, also, we are in a life-saving indication. There is no alternative treatment, so there is a highly unmet medical need. So this is currently the story of, at the end, academic research, translational research, but then, of course, the need to develop a biotech company which is due to develop the product and to organize the clinical trial for bringing this product at the end to the patient in need. And I would like to thank, of course, my hospital, Saint-Luc Hospital, my university, University of Louvain, and Cellaïon, which is the biotech company developing the product for the patients. Thank you very much for your attention.